Hello students. So in the previous class, we people have said about the steps of the recombinant DNA technology. Now in this class, actually we have started the steps of the recombinant DNA technology, and there we people have seen the three steps. Okay, the very first step is called as what? The very first step is the isolation of the genetic material. Okay, the very first step is the isolation of the genetic material from the different organisms. Second step is the what we have seen. The second step, after extracting the DNA. Okay, after extracting the DNA, whether how the DNA is cutted, that is the second step. And third step is we people have seen that amplification of the DNA by using the PCR. Okay, what we have seen? We have seen the amplification of the DNA by using the PCR technique. That we have seen in the third step. Now we are going to see the fourth step after the multiplication of the DNA. Okay, after the multiplication of the DNA, what we have to do? Okay, so see here, target DNA we have got. Target DNA is multiplied by amplification. I have told you what is amplification. That is multiplication of the target DNA into million number of copies. Multiplication of the target DNA into million number of copies, that is the amplification. So that I have told you. And here we are going to see that in the amplification is done by which process? That is PCR. PCR is the polymerase chain reaction. And also you people have seen the three steps of the PCR. One is called as denaturation, and the second step is called as annealing, and third step is called as for primer extension. What is the third step here? Primer extension. Primer extension we have seen. Okay. Now here imagine that I will draw the diagram here. Suppose imagine that this is the bacteria, and here in the bacteria there is a presence of plasmids. Okay. Now it is the target DNA. It is the target DNA. Okay, it is the target DNA. So the extraction of the plasmid from the bacteria we are going to see. It is the bacterial DNA. It is the bacterial DNA. It is the plasmid. So here what we are going to see is the extraction of the DNA. Okay, what we are going to see is the extraction of the DNA. Okay, you can see here the target DNA is inserted into the plasmid. Okay, the target DNA is inserted inside the plasmid. So this is actually one more step is the PBR322. So this is the PBR322, where by using the HIND2 enzyme, that is restriction endonuclease enzyme, we are removing the tetracycline resistant gene. Okay, and in place of tetracycline resistant gene, we are adding the new DNA molecule, or we will say the target DNA. Okay, what we are adding there? The target DNA. So two organisms DNA is added, and that is called as what? Recombinant plasmid. What do we call for this? It is called as recombinant plasmid. What do we call? Recombinant plasmid. Now this recombinant plasmid, next step is it is inserted once again inside the bacterial cell. Okay, this recombinant plasmid is inserted once again in the bacterial cell. Okay, inserted once again in the bacterial cell for cloning. For cloning. What is the meaning of cloning? Cloning means the process where the target DNA is multiplied by using the bacterial cell, and it is genetically and morphologically similar to the parental cell. Genetically and morphologically similar to the parental cell. So these cloning cells, okay, these clone cells, see here, this is the fourth step. What is that? Here the clone cells are produced. Daughter cells are produced with the recombinant plasmid or the recombinant DNA. Okay, recombinant plasmid or the recombinant DNA. So this is called as the fourth step. So which is the fourth step? Insertion of the recombinant plasmid into the host cell. Okay, insertion of the recombinant plasmid into the host cell. Now, in the biotechnology, what host cell we are using? We are using the E. coli bacteria. E. coli bacteria as the host cell. E. coli bacteria as the host cell. Okay. So, E. coli bacteria, before the insertion of the recombinant plasmid inside the E. coli bacteria, what treatment is done for the E. coli bacteria? First, the E. coli bacteria is taken. It is treated with the cold calcium chloride solution. First is it is treated with the cold calcium chloride solution. Cold calcium chloride solution. Okay. Then it is taken to the 42 degrees Celsius temperature. Then it is taken to 42 degrees Celsius temperature. Then once again back to the ice. Then once again back to the ice. Back to the ice. What happens? Small pores are created on this. Small pores are created on this. And with the help of these small pores on the cell wall, there is an insertion of the recombinant plasmid. There is an insertion of the recombinant plasmid. Okay. So here the host cell is first treated with the cold calcium chloride solution, then taken to the 42 degrees Celsius temperature, then once again back to the ice. So when it is taken back to the ice, what happens? The small pores are created on the cell wall. So this small pores, because using the small pores, there is an insertion of the plasmid. Okay, using the small pores, there is an insertion of the recombinant plasmid inside the host cell. Okay, there is a process of recombinant plasmid inside the host cell. So that is the fourth step. That is insertion of the recombinant plasmid inside the host cell. Okay, that is the fourth step. Okay. Now before insertion, what they will do? It is treated with the cold calcium chloride solution, taken to 42 degrees Celsius, then once again back to the ice they have taken. Okay. Now next is we are going to see the next step. Next we are going to see the next step, that is what to say, obtaining the foreign gene product by using the bioreactor. Okay, obtaining the foreign gene product by using the bioreactor. Fifth step, obtaining the foreign gene product by using bioreactor. By using bioreactor. Hope so till now you have understood. Okay, till now you have understood. Till here you have understood. Next is we are going to see the obtaining the foreign gene product by using the bioreactor. So before going to go these steps, just you do the revision of all the steps, then we'll go for this concept. Well students, this is the fifth step, that is obtaining the foreign gene product by using the bioreactor. Now before going to this step in detail, we have to see that what is the use of the bioreactor and how many types of bioreactor and structure of bioreactor we have to see now. So first of all, before going to this, we have to understand what is bioreactor. Bioreactor is a large stainless steel container. Bioreactor is a large stainless steel container which is consisting of 100 to 1000 liter of volume. 100 to 1000 liters of volume. Okay, that means whichever product we want to synthesize, we have to use the bioreactor. We have to use the bioreactor, and what is the uses? It can reduce the primary metabolites. It is having the capacity to synthesize the secondary metabolites. Okay, organic acids in a larger amount. Understood? All these are utilized in the medicinal purpose. Okay, why all these are synthesized in a larger amount? Because it is used in the medicinal purpose. So for that process, we are going to see the next step as the obtaining the foreign gene product by using the bioreactor. Now see here, bioreactor are of two types. This type of bioreactor is called as simple stirred tank bioreactor. This type of bioreactor is called as what? Simple stirred tank bioreactor. Simple stirred tank bioreactor. And this type is called as what? Sparked stirred tank bioreactor. This type is called as what? Sparked stirred tank bioreactor. Sparked stirred tank bioreactor. Okay. Now see here, this one is the simple stirred tank bioreactor. This one is the sparked stirred tank bioreactor. Now it is a cylindrical shaped structure. Okay, having a long tubular shaped structure support and tube-like structure, and it is a plate. Okay, and here is the motor. Motor is attached with the help of the support called as neck, and these are called as impeller. Flat blade impeller. Spoon will be done for mixing. Like the spoon, they will be active. Okay, in anti-clockwise rotation, they are used to rotate. Okay. Now here you just we will put the host cells inside this. Okay, we are putting the larger amount of the host cell inside this. Then along with that, we are adding the chemicals also. Along with that, what we are adding? We are adding the chemicals also. So what happens? The host cell and the chemicals, okay, host cell and the chemicals are mixed rigorously. Rigorously means what? Very speed. Okay, they are mixed rigorously, and then the product is synthesized there. And then the product is synthesized. Okay, and here we are going to see that after the synthesis of the product, okay, after the synthesis of the product, we are going to see that the product when it is synthesized, okay, the product when it is synthesized, we are going to see the synthesis of the new product along with the some waste material, So this waste material has to be removed. Now, for example, I will give one small example. Insulin is a medicine for the diabetes patient. Okay, to synthesize the insulin in a larger amount, what we will do? We will take the insulin gene from the human beings. Then after taking the insulin gene from the human beings, we will insert that in the plasmid. It will become recombinant plasmid. That recombinant plasmid, we are inserting that in the host cell, that is in the E. coli bacteria. You people know that by the E. coli bacteria, by undergoing the binary fission, it is producing two daughter cells, from two daughter cells to four daughter cells, from four daughter cells to 6 to 8, like that it will produce. So as the we'll take the host cell and we'll keep that in the bioreactor. What happens? The insulin gene is synthesized. Imagine that this is the insulin gene and it is attached with the some unwanted material. Now we want to remove this unwanted material. Okay, what we have to do? We wanted to remove this unwanted material. So what we'll do? What we'll do here? That is we will take some chemicals and the enzymes. We are taking some chemicals and the enzymes, and then we are removing the unwanted material. Okay, and then here the pure insulin will get. Okay, so same process is done here also. So the culture broth is nothing but bacteria are added, chemicals are added, and it is switched on. It is mixing rigorously. Okay, it is mixing rigorously, and then the product is ready. And that product is taken from this point Okay, and it is checked whether it is neat or not, or it is attached by any of the unwanted material. Then what they will do? They will remove the unwanted material by using the enzymes, anti-enzymes we will say. By using the anti-enzymes, there is a removal of the unwanted material. So maximum in the medicinal industries, in the pharmaceutical industries, these bioreactors are utilized. Maximum in the medicinal industries, in the pharmaceutical industries, these bioreactors are utilized. That is simple stirred tank bioreactor, and this simple stirred tank bioreactor is very important for the final exam also. It is very important for the final exam also. Now, sparked stirred tank bioreactor. It is also one type of bioreactor. Sparked stirred tank bioreactor is also one type of bioreactor where it is somewhat container shaped structure. It is a container shaped structure, and it is what it is doing. It is also helping for the production of more amount of products, large amount of products we'll say. Okay. The difference here is it is used in the small scale industries. The simple sparked stirred tank bioreactor is used in the small scale industries. The simple stirred tank bioreactor is used in the large scale industries. Okay. Once again I'll repeat, concentrate here. Bioreactor are the large stainless steel container which is having the 100 to 1000 liters of volume in size. And this bioreactor is used to synthesize the primary metabolites, secondary metabolites, organic acids in a larger scale. Understood? And we are going to see that these bioreactors, okay, these bioreactors are of two types. One is called as a simple stirred tank bioreactor, and the other one is called as what? Sparked stirred tank bioreactor. Usually, the simple stirred tank bioreactor is used in the large scale industries. Okay, in the large scale industries, the simple stirred tank bioreactor is utilized. In the small scale industries, the sparked stirred tank bioreactor is utilized. Now, if you see the parts of the bioreactor, it is a cylindrical shaped structure having a culture broth, motor, and also we are going to see the flat blade impeller spoons we are going to see for the mixture of the culture. That means for the mixture of the content produced in the bioreactor. Okay, and these are utilized to synthesize the products in a larger amount, especially in the case of pharmaceutical industries. Okay, and this is very important for the 5 marks for the final exam. 5 marks concept it is very important for the final exam. So here in the recombinant DNA technology diagram also very important for 5 marks. It is a diagram of the recombinant DNA technology which is also very important for the 5 marks. Now we are going to see the last step of this recombinant DNA technology, that is called as what? Downstreaming process. Here itself I will write here. Okay, so that you can write down all the points what I have written on the board. Okay. Last step is the downstreaming process. Okay, what is the last step here? Downstreaming process. Okay, downstreaming process. What is this downstreaming process? It is the process. Okay, it is the process of. It is the process of separation and isolation of the product. Separation and isolation of the product. It is the process of separation and isolation of the product. I have told you already, when the product is synthesized, it is not in the pure form. Okay, when the product is synthesized, it is not in the pure form. That has to be converted into pure form. How it is converted? By the removal of the unwanted material attached to that product. Okay, that is removed by using the chemicals, and then it is separated from the original form. That is called as what? That is called as downstreaming process, which is the last step of the steps of recombinant DNA technology. Which is the last step of the recombinant DNA technology. So in the recombinant, so the totally Totally, the recombinant DNA technology was the last concept of the biotechnology principle and process. So once again I'll repeat, please concentrate. Recombinant means the combination of the two organisms DNA. That is called as what? Recombination. And here we are going to see the steps of the recombinant DNA. First is isolation of the genetic material from the different organisms. That is, if you want to extract the genetic material from the bacteria, then what you will do, you will use the enzyme called as lysozyme. Okay, you are using the enzyme called as lysozyme to disintegrate the cell wall of the bacteria. Suppose in case if you want to extract the genetic material from the fungi, what you are doing, you are using the chitinase enzyme for disintegrating the cell wall of the fungi. Suppose if you want to extract the DNA from the plant cell, what you are doing, you are utilizing the cellulase enzyme to disintegrate the cell wall component of the plant cell. Then the DNA is coming outside. After coming the DNA, that means after coming outside, the DNA is attached with the RNA and the protein. So to remove the RNA, which enzyme is utilized? Ribonuclease enzyme is utilized. To remove the protein, which enzyme is utilized? Protease enzyme is utilized. Then we are getting the pure form of DNA, which is stored in the chilled or cooled ethyl alcohol. Then coming to the second step here, second step says that fragmentation of the DNA by using the restriction endonucleases enzymes. So REN is also called as what? Molecular scissors or molecular knives we will say. And after taking out the DNA is cut into small pieces. Okay, the DNA is cut into small pieces. And those pieces are taken for the further procedure, that is amplification of the PCR. That means amplification of the target DNA by using the PCR. Suppose if we want to multiply the fragment of the DNA into million number of copies, then we have to go for the process called as amplification, which is a third step of the recombinant DNA technology. What is amplification here? That is the multiplication of the DNA into million number of copies. Okay, without changing the sequence. Sequence change ಆಗಲಾರ್ದನೆ ಅದನ್ನ ಮಲ್ಟಿಪ್ಲೈ ಮಾಡಲಿಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೀವಿ? ಆಂಪ್ಲಿಫಿಕೇಶನ್ ಆಫ್ ದ DNA ಅಂತ ಕರಿತೀವಿ. ಆಂಪ್ಲಿಫಿಕೇಶನ್ ಯಾವುದು ಪ್ರಾಸೆಸ್ ಇಂದ ಆಗ್ತದ? ಇಟ್ ಇಸ್ ಬೈ ಯೂಸಿಂಗ್ ದ PCR. PCR ಅಂತಂದ್ರೆ ಏನು? Polymerase chain reaction. Okay, after so many days I am using the Kannada words here. Okay, what is PCR? That is polymerase chain reaction, which has been synthesized by the scientist called as Gary Mullis. Okay, Gary Mullis is the name of the scientist who has synthesized the PCR, and this PCR is also called as thermocycler machine. PCR vinodena ಹೆಸರು thermocycler machine ಅಂತ ಕರಿತೀವಿ. And here we are going to see the presence of three steps. ಮೂರು ಸ್ಟೆಪ್ಸ್ ಇದಾವೆ PCR. ಒಂದು ನಾವು ಡಿನ್ಯಾಚುರೇಷನ್ ಅಂತ ಕರಿತೀವಿ, ಇನ್ನೊಂದು ಅನಿಲಿಂಗ್ ಅಂತ ಕರಿತೀವಿ, ಮತ್ತೊಂದಕ್ಕೆ ನಾವು ಪ್ರೈಮರ್ ಎಕ್ಸ್ಟೆನ್ಷನ್ ಅಂತ ಕರಿತೀವಿ. ವಾಟ್ ಈಸ್ ದ ಮೀನಿಂಗ್ ಆಫ್ ದ ಡಿನ್ಯಾಚುರೇಷನ್? ಡಿನ್ಯಾಚುರೇಷನ್ ಮೀನ್ಸ್ ವಾಟ್? ದಟ್ ಈಸ್ ದ conversion of the double stranded DNA into single stranded DNA fragments. Okay, conversion of the double stranded DNA into single stranded DNA fragments, that is called as what? Denaturation, and it is occurring at the temperature that is 94 degrees Celsius. Okay, usually 92 to 94 degrees Celsius. Then second step is annealing. Annealing means joining of the oligonucleotides at the temperature 42 degrees Celsius, and here the TAC DNA polymerase is helping. TAC means what? Thermus aquaticus. Okay, name of the bacteria which is synthesizing the polymerase enzyme. Then next is we are going to see the concept called as primer extension, where 3' and 5' end are joining to this process. That is the primer extension, and it will occur at the temperature 70 degrees Celsius. Okay, 70 degrees Celsius temperature is required. Above 70 degrees Celsius, TAC DNA polymerase is not working. Okay, next step is insertion of the recombinant plasmid inside the host cell. So before the insertion of the recombinant plasmid inside the host cell is treated with the cold calcium chloride solution, then taken to the high temperature, that is 42 degrees Celsius, and then once again back to the ice we are going to see. And there after that we are going to see the creation of small pores on the cell wall, in which we are going to insert the DNA. Okay, in recombinant plasmid we are inserting that. But next, after that we are going to see that after the fourth step we are going to see the fifth step, that is obtaining the foreign gene product by using the bioreactor. Bioreactor are the large stainless steel container which is consisting of 100 to 1000 liters of volume, okay, which is consisting of 100 to 1000 liters of volume. And here we are going to see the presence of bacteria, okay. We are going to see the presence of the bacteria we are adding, chemicals we are adding, then we are adding the other chemicals, okay, so that the product is synthesized that. Okay, so that the product is synthesized that, and the synthesized product will be having a waste material. So we have to remove that waste material. To remove the waste material, certain chemicals are utilized, okay, and then the product is synthesized there. That is, to synthesize those products, we are using two types of bioreactor. One is called as simple stirred tank bioreactor. Other one is called as sparked stirred tank bioreactor. So simple stirred tank bioreactor is a large stainless steel container. It is a large stainless steel container which is having the 100 to 1000 liters of volume, and it is useful for the synthesis of the products in a larger amount. Okay, then here in the final exam, the simple stirred tank bioreactor is very important concept for you to draw the diagram, and they will ask the explanation also. And here spark stirred tank bioreactor we are seeing. So it is just like a container what we are going to see, where it is also helping for the large synthesis of the products. Okay. Now after this, students, next step what we have to see is downstreaming process. So downstreaming process means what? Separation and isolation of the DNA fragments. Okay, separation and isolation of the DNA fragments is called as what? Downstreaming process. That means final product has been synthesized there, but along with the final product, certain unwanted materials are attached there. So that to remove that unwanted material, certain chemicals are utilized and then it is separated. That is the final product, you will say, and that process is called as what? Downstreaming process, which is asked for 1 mark question. Okay, so this completes your chapter called as biotechnology principle and process. Okay, this completes your chapter, that is biotechnology principle and process. For your next test also, I am giving this chapter for the CET test as well as assignment question. Okay, and here we are going to see that one more concept is there. In this chapter, one more concept is that there is gel electrophoresis. That is also very important for 5 marks. Gel electrophoresis is also very important for 5 marks. You can see the OBS class, that gel electrophoresis. Once again, I'll just revise that gel electrophoresis. Gel electrophoresis is a technique. It is a technique to separate and isolate the DNA fragments. And here we are going to see that the large tray is required, buffer solution is required, agarose gel is required, then shotgun is required, switch is required, everything is required here. Okay. Then what we will do, agar powder will take. From where do we get the agar powder? From the algal organism called as Gracilaria and the Geladium. Okay. Gracilaria and Geladium are the algal components which is releasing the agar powder. Agar powder is taken and it is mixed with the water, distilled water it is mixed, and then the agarose gel is prepared. Okay, and after the preparation of the agarose gel, that the gel is added in the tray. Okay, then it is cooled and then the large teeth comb is taken, inserted inside that, and the wells are created. And when the wells are created, in that wells we are going to see that there is an addition of the DNA. Okay, and switch is on, the DNA is transferred in the form of bands. Initially the larger bands are produced. Okay, initially what? The larger bands are produced, and later on the smaller bands are produced there. Okay, later on smaller bands are produced. So this completes your gel electrophoresis. Along with that, we are completing the chapter called as biotechnology principle and process. In the next class, I will start the new chapter, that is biotechnology and its applications. Now write down all the points and all the diagrams, whatever it is there on the board. This is the diagram of the recombinant DNA technology, which is very important for 5 marks. These are also very important for 5 marks. Write down this and draw this diagram.